This is podcast number 44. We're going to start talking about guard cells, and this is continuing on in homeostasis of plants, more at the whole organism or tissue level rather than at just the cell level. If you look at the surface of a leaf with a microscope, you see these kidney-shaped cells, bean-shaped cells that surround an opening. The opening is called a stoma. Stoma is, I think it's Latin for mouth or hole or opening. So the stoma is the opening, and the cells surrounding the stoma are called guard cells. Plural of stoma is stomata. So this is how gases exchange with the inside of the cell in a plant. So we talked about during photosynthesis how CO2 from the atmosphere is fixed during the Calvin cycle. That CO2 has to enter the plants through stoma, and these guard cells will change shape to make this opening big or small. In addition to CO2, it's got water vapor that can be lost. So you would predict that during bright light, when photosynthesis is really cranking, you would want your stomata to be open to allow CO2 in for carbon fixation. If conditions were under high humidity, you would also want those stomata to be open because the air is humid, they're not going to lose water vapor. But if the conditions are dry and the plant will tend to lose water through their stomata, you would want to close those stomata to prevent water vapor loss. Of course, if you close the stomata, it's very hard to get CO2 in there. There are hormones that affect whether or not the stomata are open or closed. So if you take stomata and you prepare those cells outside the leaf, and you just measure how big is the stoma, how big is the hole surrounding that the guard cells surround. If you don't treat them, it's a big hole. Treat them with abscisic acid, which is a plant hormone, closes the hole. Abscisic acid plus a nitric oxide scavenger. Nitric oxide is a gas that acts like a signaling molecule, and these scientists suspected that nitric oxide was involved in opening and closing the stomata, so they put in a nitric oxide scavenger to suck up all, like a sponge for nitric oxide, and they saw that it does not, if there's no nitric oxide around, they're not as closed as with the abscisic acid than if there is nitric oxide around. If you provide extra nitric oxide, they will close off. And if you put both extra nitric oxide and a scavenger, so you give it extra nitric oxide and a way to suck it up, they are mostly open. So it's a combination of the effects of the hormone and nitric oxide that govern the opening of those cells. Looking at what the cells themselves, they used a fluorescent dye to indicate if nitric oxide was there or not. The fluorescent dye changed color if nitric oxide was there or not. So if they don't treat it with abscisic acid, they get red, which indicates no nitric oxide. But if they do treat it with abscisic acid, they get green. Green indicates, yes, there is nitric oxide there. If they add the scavenger, it sucks up the nitric oxide. It sucks up most of the nitric oxide, not all of it. You can see most of the green goes away. And if they just add a dye that is not sensitive to nitric oxide, they don't see much of a change. And this was just to show that the dye itself wasn't causing the cells to open or close. Okay, so this correlates with an open cell, a mostly closed cell, mostly open cell, mostly open cell. It can be kind of hard to tell the difference between open and closed because they don't close completely flat. They're always a little bit open. You can also do electrophysiology on plant cells, just like you did electrophysiology on the neurons, where you measure ions moving in and out of these guard cells. And does that have anything to do with how they open or close? So if you measure the resting membrane potential, just what's the inside relative to the outside, it's negative. Okay, just like in neurons, it's negative. And if you shine light on these guard cells, the membrane potential becomes more negative. And then if you shut off the light, it becomes less negative. So it could be becoming more negative because negative ions are pumped into the cell. It could be becoming more negative because positive ions are coming out of the cell. If you look at the characteristics of the light and how it affects the guard cells, only blue light matters. So if you shine a red light on it, nothing happens. Green light on it, nothing happens. Only blue light causes something to happen. And more blue light causes a bigger effect than less blue light. Okay? If you see how stomata respond to CO2. So CO2 is what's needed for the Calvin cycle. CO2 is going to enter through the guard cells. And so what you would expect is that if there's a bunch of CO2 around, then you don't need more. And the aperture, the stoma, should get smaller. And that's what happens. So in this period here of this graph where you increase this concentration of CO2, that closes off the stomata. Okay? And that is associated with sucrose production. Sucrose is made as a byproduct of carbon fixation in the Calvin cycle. So this is a picture of an open stomata, closed stomata. Again, they don't close all the way, but you can sort of see that the hole is, for gas to go through, is bigger here than it is here. And here are the guard cells on either side of it. How do those guard cells change? So it's a combination of a change in water going in and out of those guard cells. So water sort of pumping them up. Remember, plant cells have a cell wall, and if you push water into plant cells, they'll get really, really stiff because of that cell wall. And also a rearrangement of the actin cytoskeleton. So actin, the same molecule that causes that myosin pulls on in skeletal muscle in animals, that same molecule actin forms part of the cytoskeleton that gives these guard cells their shape. And that actin will rearrange in an open cell versus a closed cell. And that helps change the shape of this guard cell. So if you put your hands together like you're holding a ball with your fingertips touching each other and the heel of your hand touching each other and a ball in the middle, you can think of that as a guard cell. And if you contract your muscle or relax your muscles so that your hand is flat, that would be a closed guard cell. And then if you put your fingertips together like it's holding a ball, that would be an open guard cell. So the actin rearranges itself to make those two different shapes, and that will govern open versus closed. So there are lots of things that affect whether or not stomata will be open. We talked about a hormone a plant hormone, abscisic acid, that could affect it. We talked about nitric oxide, which is a gas that is a signaling molecule that can affect it. We talked about the actin cytoskeleton rearranging. We talked about ions moving. I didn't tell you, but the ion that is moving in and out of these cells that is measured by these electrodes is potassium. And it's actually potassium is moving out that causes the membrane potential to go down. So potassium moves, and then light can also affect it, but only certain colors of light. And then not only that, but plants also seem to have coordinated movements of their stomata. So they don't have a neural network like animal cells do. They don't have neurons, and neurons can coordinate the response of stimulated cells if they're in contact with that neuron through a synapse. But plants don't have neurons. So how would one cell decide to close? There seems to be, this is really hard to see in this diagram, and there was a movie in your book to show this, but the link is dead, and I contacted the authors, and hopefully they're going to fix that soon. Hopefully by the time you see this, they'll fix that movie. But in a black and white picture, they can tell the difference between a closed stomata and an open stomata by this difference in gray. And I admit this is really, really hard to see. But then they color-coded those using a program like Photoshop, and then they could track recently closed stomata and color those yellow, recently open stomata, color those red, and then stomata don't change and color those black. And then track those over time. So this was recently closed, and then it's recently opened over time, and then it's going to close again over here. And from this sort of setup, this sort of system where they're watching stomata under different conditions, what they could show was that across an area of leaf, like from the top to the bottom, is that stomata tend to pay attention to their nearest neighbors. So if the stomata around you are closing, it's more likely you will close also. And if the stomata around you are opening, it's more likely you are going to open also. There are connections between plant cells, little connections between the cytoplasm of one plant cell and another plant cell, and that may be a signal from one set of guard cells to another guard cell that makes it that they get the message saying that your neighbors are closing or your neighbors are opening. But that's still actively being researched. So the idea here is just that plants are reacting to their environment. They can react to their environment by opening or closing their connection to the environment, the gas in their environment. The gas in their environment is critical to the plant's physiology because it's how they make food. But they have to be careful because if the air is too dry, they can lose all their water and the plant can wilt. So there are many mechanisms that are complex that govern whether or not these stomata are open or closed.